From feedstocks to fuels, tracing the CABBI pipeline. The Center for Advanced Bioenergy and Bioproducts Innovation, or CABBI, is one of 4 bioenergy research centers funded by the Department of Energy. CABBI's mission is to develop the scientific and technological basis for efficient and productive bioenergy crops, whose biomass can be converted into valuable bioproducts in an economically and ecologically sustainable fashion. In 2019, CABBI researchers planned a project to demonstrate the center's full feedstock to fuels pipeline. This consists of propagation, field trials, harvest and transport, juicing, juice fermentation, and finally, bagasse pretreatment, hydrolysis, oil recovery, and fermentation. The feedstock propagated for this demonstration was oilcane, a type of sugarcane with elevated lipid content developed during the Petros project, which was funded by ARPA-E within the Department of Energy. Researchers at the University of Florida cultivated a wild-type sugarcane in 2 lines of transgenic oilcane to be compared with one another throughout the project. Field trials were conducted at 3 locations: the University of Florida, the University of Illinois Energy Farm, and Mississippi State. A CABBI team at Mississippi State assisted in propagating the plants, sampling throughout the development. CABBI partners at Brookhaven National Laboratory and the University of Nebraska-Lincoln were instrumental in conducting analysis of these samples to test for oil content variation. The harvested material was split to be used to produce subsequent generations for future experiments or to be processed immediately at the Integrated Bioprocessing Research Laboratory, or IBRL, at the University of Illinois. The movement of this transgenic material was governed by USDA APHIS permits after hundreds of kilos were packaged into bags, loaded on a freezer truck, and delivered to be processed. A juice press at the Illinois Energy Farm, followed by a fiber press at IBRL, were used to generate sugar-rich juice in dry, fibrous bagasse. CABBI teams at IBRL, Mississippi State, Brookhaven National Laboratory, the U.S. Department of Agriculture, and the University of Nebraska analyzed the juice in bagasse for sugar content, oil quantity, and the location of the oil within the plant itself. A portion of the juice underwent immediate fermentation. In this process, a yeast consumed the sugars present in the juice and produced bioethanol. As new and improved engineered yeasts are developed in CABBI, they'll be evaluated in the fermenter, and the material will be further processed by IBRL to isolate the valuable co-products. Some of the juice did not undergo fermentation immediately, and was instead set aside for further testing by researchers in CABBI's conversion theme. They evaluated the juice's suitability as a growth medium for various microorganisms and checked for the inhibitors within the broth, which could generate a harsh environment and hinder fermentation. The fibrous oilcane bagasse was ground, washed, and dried to an optimal moisture before undergoing pretreatment, a process used to make fermentable sugars more accessible for yeast to convert into bioethanol. The bagasse was pretreated through a combination of careful feeding, heat, and pressure in the IBRL's unique continuous pretreatment reactor, helping release the fermentable sugars and preparing the material for enzymatic hydrolysis. The hydrolysis step was performed in a temperature-controlled, agitating reactor to ensure optimal conditions for the enzymes to release sugars during fermentation. After hydrolysis, all remaining oils and solids were removed from the mixture, and the resulting broth underwent fermentation. During bagasse fermentation, the yeast consumed the sugars to produce oil instead of ethanol. When the fermentation process was complete, the cells were harvested and lysed, and the oil was collected for analysis and processing. The capabilities of the CABBI team to engineer, grow, handle, process, and evaluate grasses producing oils for bioenergy are unparalleled. We will continue to use this pipeline approach to evaluate new feedstock lines and yeast strains to optimize creation and recovery of valuable bioproducts.